much. And again, Tama, thank you for the invitation. This is I think, the 5th time I've been here, and really do enjoy coming here and catching up with old friends and talking about the work we do. Can you hear me if I just walk around? I hate to stand behind a lectern. Okay, excellent. So what I thought I'd like to do to you is today is just bring you up to date with some of the work. I'm a basic scientist, so I really ask fundamental questions related to this disease that we are all so very interested in, and this very enigmatic disease. So what I'm going to do is take you through a little journey, and hopefully I can get that done in 15 minutes, about some of our more recent work and trying to understand the implications of microRNAs and their role in the pathogenesis of this disease. So the question, of course, is, what kind of model can we use? We all know that by the time diagnosis is made in women with disease, it's been there for an extended period of time. So it's very difficult to ask fundamental biological questions about what initiates this disease, how does this disease impact both pain and infertility. And so we have to come up with appropriate model systems. And several years ago, my laboratory, along with Thomas de Hoog's laboratory in Belgium, developed the baboon as a non-human primate model. So this is an old world monkey. They get spontaneous disease. Their physiology, anatomy, and endocrinology is very, very similar to the human female. So because of the phylogenetic relationship and the women, it allows us to utilize this model to try to understand the fundamental aspect of this disease. So basically, we use this animal model to essentially create a peritoneal disease with the implications of retrograde menstruation. So essentially what we do with these animals is once we evaluate them laparoscopically, because they do get spontaneous disease, and make sure they have no spontaneous disease, we inoculate the peritoneal cavity with menstrual tissue on 2 consecutive menstrual cycles. And with that, these animals will develop full-blown disease that's very, very similar to what you see in the human situation. So this is a baboon that has got endometriosis 3 months after we've inoculated these animals with menstrual tissue, and you can see how similar the peritoneal disease is to the human female. The advantage of this animal model is that we can then sample the same animal prior to and after the induction of the disease. So we know from day 1 when we inoculated these animals when the disease began, and then we can follow the progression of the disease in these animals. And we've utilized this model for several years, and essentially what we've been able to show is that, since our interest is really the impact of the disease on infertility, is that progesterone resistance, which is now considered one of the hallmarks of this disease, occurs sometime about 6 to 9 months after the disease is initiated. And what you see basically is that you see a very early response once you inoculate these animals in terms of the gene expression of the endometrium, which has a very estrogenic response in the mid-secretory phase. So we always focus on the mid-secretory phase because that's the window of implantation, the time at which the embryo attaches to the endometrium. And then subsequently, somewhere between 6 and 9 months, the endometrium changes and becomes what we call progesterone resistance, in that the normal genes that are regulated by progesterone during the mid-secretory phase that we think might be important for the initiation of pregnancy are all affected. In addition, when we look at the global changes, so using Affymetrix and various gene arrays, what we were able to also show that 1 month after we initiate the disease in these animals and we look at the endometrium using global gene expression technologies, that 40% of the global changes that we see in these animals at 1 month are comparable to what's being reported in women who've had the disease for the extended period of time. So essentially what this is telling you is that the early insult that you see in the peritoneum is having a marked effect on the endometrium. So we've done a lot of this work, and what I wanted to really focus today is trying to ask the questions in terms of what is the upstream mechanisms that might be involved in some of these processes that are responsible for contributing to infertility that's associated with this disease. So based on all of this, our question went back then to try to understand whether microRNAs play a role in this process. And essentially what microRNAs are is they are small 17 to 24 base pair single-stranded RNA molecules that have a profound effect on gene expression. And essentially, just to give you a very small primer on what microRNAs are, basically pre-microRNAs that are processed within the nucleus are exported through very unique mechanisms into the cytoplasm, and they undergo specific processing processes where the mature microRNA complexes with this RNA silencing complex. And essentially what this does is binds generally to 3' regions of the RNA and essentially acts as a brake. So unlike we think about normal transcription where either epigenetic or transcriptional regulation brings about the result of microRNAs which then translate into protein that have function, which is the classic way of looking at what happens during gene regulation, microRNA regulation actually acts more as a brake. So essentially it does the opposite of what we think of normal transcriptional regulation, in that microRNAs in general tend to suppress protein expression and thereby function. So essentially with microRNAs, it was one of the breakthroughs of the year in 2002. And just to give you a feel for how this field has just exploded, this is just a publication just using simply the title microRNA using PubMed. And you can see from its initial discovery, which was 2002 when it was the molecule of the year, you can see this precipitous increase in the number of papers that have been published in the last 10 to 12 years with regards to trying to understand the role of microRNAs as it relates to biological function. So when it comes to endometriosis then, there have been a number of papers that have described microRNAs that have been isolated, described, and identified with regards to microRNAs and endometriosis. And since the first report related to microRNAs in endometriosis in 2007, PubMed now shows between about 92 and about 100 papers that are associated with microRNAs and endometriosis. But the question still remains whether this is a cause or consequence of this disease, that we see these changes in microRNAs, which is always a question we always have in endometriosis because it takes so long to diagnose the disease. And the second question is, what do these microRNAs do if they are being regulated? How do they work? And how do we identify target genes that we can potentially target or suppress, depending on what we want to do with regards to treatment and therapy of this disease? So what I'm going to take you through is how we've approached these questions and how we've been trying to, in a very short period of time, trying to address these issues with regards to cause and consequence and function of these microRNAs. So going back then is how are these being regulated? So again, we have the advantage of our baboon model, where essentially we can use human platforms. So that's the advantage, again, because of the close phylogenetic relationship, we can do direct comparisons between human and non-human primates. And essentially what we did in this initial experiment was took endometrium from animals prior to the induction of the disease and 3 months after the induction of the disease, and using an adjuvant array, try to identify the microRNAs that were altered in response to the disease, doing a direct comparison in the same animal model. And what we basically find is that there are a number of microRNAs that are significantly altered. The ones in bold are the ones that have also been reported to be altered in women. So here again, you can see within 3 months of the induction of the disease, using peritoneal inoculation of menstrual tissue, you can see the microRNAs that are very, very similar to what has been already reported in women. And for the purpose of today's talk, I'm going to focus on 3 of these. microRNA 451, which is highly downregulated in response to disease, and 29C and 21, which are upregulated in response to disease. So what I'm going to do is try to take you through how we try to approach what the potential mechanisms and functions of these microRNAs are. So looking through the literature, the decrease in microRNA 451, which has been extensively studied in breast cancer and hormone-resistant breast cancers, the decrease is also seen in breast cancers is associated with the enhanced proliferation and invasion, which again, as you know, is a characteristic of endometriosis. microRNA 29C, which is upregulated, targets one of the genes. It targets a number of genes. That's the one thing with the microRNAs. They're complex. They target multiple sometimes thousands of genes. So one microRNA can have lots of regulatory mechanisms. But the one we've been interested in was specific immunophilin called FKBP4, which is a target of microRNA 29C, which we think contributes to the progesterone resistance of the disease. And our very recent work in looking at microRNA 21, which is also highly upregulated in breast cancer, and it promotes fibrosis, inflammation, and EMT, again, mechanisms that we know are associated with this disease. So I'm going to take you through very quickly in terms of how we try to approach these and try to see whether there are functional implications associated with this disease. So first and foremost, when you do any kind of array, you want to make sure that that array validates what you're seeing by looking at the tissue itself. So this is the 451 expression, which is the one that's markedly downregulated. And you can see then in the baboon model, progressively, as once a disease is induced, you see a dramatic downregulation across the timeline in the same animals, same group of animals, between 1 and 15 months, along with the lesions that we then harvest at 15 months. And if you then look at women with this disease, you see the same thing. Utopic endometrium with women with endometriosis compared to controls, again, all in the mid-secretory phase. You can see the dramatic downregulation of 451, which complements what we see in our induced model. And then, of course, looking at targets, one target we've been really interested in looking at is YHAS, a 14.3.3 zeta, which is a gene that's involved in regulating proliferation. And what you can see is, just as you would expect, when a microRNA goes down, its target gene goes up, and you can see that the expression of this gene, both in the utopic endometrium and the ectopic endometrium, is markedly increased. And the one thing you have to do with all microRNA studies is to validate this target. And essentially what you do is you can then use your target gene with a luciferase construct, which gives you a readout. And essentially, if you overexpress 451, you can see that the gene expression based on luciferase activity, which is a readout of light activity, goes down. But if you essentially mutate that region that 451 binds to on the target gene, you don't see an effect. So that tells you that it's a bona fide target of the microRNAs. And we think that this gene, based on the literature, is also associated with proliferation. And in women with this disease, Juan Garcia in Madrid has shown that this particular gene also is the protein is upregulated in the uterine flushings of women with this disease, and it increases as the progression and the severity of the diseases increases. So basically, based on the breast cancer literature and also work that's been reported in women, we wanted to see whether this microRNA plays any role in proliferation. So we first went back and looked at our baboon endometrium. And so these are the control endometrium, looking at the in situ hybridization of microRNA 451. And you can see in the control animals, you see very beautiful localization of the microRNA by in situ hybridization in the epithelial cells, which is completely eliminated in the animals with this disease. And then if you look at the target, which is YHAS, which interacts with beta-catenin and YAP, which are basically regulatory genes that are involved in proliferation, these genes are not expressed in the endometrium of animals that do not have the disease. However, in contrast, if you look at the expression of YHAS, beta-catenin, and YAP, they all are markedly upregulated in the utopic endometrium and also in the ectopic endometrium that I'm not going to get into today when 451 is downregulated. And this is correlated with a significant increase in proliferation, which when you use a phosphohistone 3, which is a marker of mitotic activity, you can see that there's a significant increase in the expression of proliferation in the mid-secretory phase when epithelial cells should not proliferate in the endometrium, suggesting that this change in microRNAs is involved with this aberrant proliferation that we see in the utopic endometrium. And you can go ahead and then confirm this by simply taking cell lines, and in this case what we've used is an epithelial cell line. Essentially, when you overexpress 451, you downregulate both messenger RNA and protein for YHAS, and this is correlated then if you do an MTT assay and look at proliferation, there's a significant decrease in the proliferative activity of these cells when you overexpress 451, suggesting that this could potentially be an important mechanism by which the proliferation takes place aberrantly in the utopic and ectopic endometrium associated with this disease. So I'm going to summarize a huge amount of work here in this one diagram. And essentially what we see is that microRNA 451 has two major targets that we've been really interested in, the cell cycle targets, the cell cycle genes, and YHAS. And essentially the regulation of these contributes through YHAS and beta-catenin and interaction of beta-catenin and YAP being translocated to the nucleus, enhances cell survival, promotes invasion, and C-FOS, if you remember from the first slide I showed you, which is highly upregulated in the utopic endometrium, interacts with YAP to enhance EMT. In addition, YHAS, together with beta-catenin and the other cell cycle genes, enhances the proliferative index associated with this tissue. So essentially what you're seeing is an aberrant change in microRNA 451, which then enhances cell cycle activity and proliferation activity. So a lot of our work right now is focused on targeting 451, since we know that if we overexpress it, we can suppress proliferation, and we are using nanoparticle technologies and a number of other methodologies to try to target 451 and see if we target lesions in various animal models and cell in vitro experiments, whether we can suppress this proliferation response. So now switching very quickly to this idea of progesterone resistance, if you look at FKBP54 now, FKBP4, you can see now both in the baboon and the human, in the normal utopic endometrium, you see a significant increase in immunolocalization of this protein that's important for progesterone action. And again, in the mid-secretory phase of both baboons and women with this disease, you see that this is dramatically downregulated. Again, confirming that there is an association, and in this case, 29C goes up dramatically in response to the disease. It's increased more markedly when progesterone resistance is dominant in this tissue, and then this is associated again with a significant decrease in FKBP4 again later on in the disease process when progesterone resistance is markedly evident in this tissue. And again, you can predict that by now using a 29C mimic, using an FKBP4 luciferase component, and showing that it's a bona fide target. And here again, since this is required for progesterone signaling, we can go back and use now stromal cells. And these are stromal fibroblasts, which we know do not undergo destabilization appropriately in response to endometriosis, that if you overexpress 29C, you downregulate FKBP4. And if you look at progesterone target genes such as decrin and HOXA10, which have all been shown very clearly to be downregulated in endometriosis, you can suppress this, along with the destabilization markers, IGF-BP1 and prolactin, suggesting that basically the upregulation 29C, perhaps through FKBP4 and other mechanisms, suppresses the ability of these cells to respond to progesterone and undergo destabilization. So the working hypothesis right now, what we have, is that normally what you need is FKBP4, which complexes with heat shock protein 90, the progesterone receptor, and the ligand forms this complex in the cytoplasm, which is required for translocation in the nucleus to initiate transcriptional regulation of progesterone responsive genes. However, in the presence of endometriosis, when FKBP4 is low because of increased levels of 29C, this complex doesn't form appropriately, so as a result, you're not able to initiate progesterone regulated transcription, and as a result, the endometrium is no longer responsive appropriately to progesterone signaling, which is so critical for the establishment of pregnancy. So again, what I've tried to show you is that in the absence of endometriosis, when we look at these two microRNAs that I focused on initially, the increase in 451 and the decrease in 29C is associated in a situation where there is no proliferation and apoptosis and progesterone responsiveness. So as a result, there is a decrease in potential lesion development, inappropriate proliferation of the uterus, which then, because progesterone then can be responsive to the endometrium, that pregnancy can ensue in the situation where we're looking at infertility. However, in the presence of endometriosis, now you have the opposite effect, where 451 is significantly downregulated and 29C is upregulated. And as a result, their target genes, and we are just focusing on 2 of the target genes, there's multiple target genes associated with that, alters proliferation and decreases apoptosis in response to the interaction with beta-catenin and YAP. And in this case, the ability of progesterone receptor to be translocated to the nucleus results in the ability of these lesions to develop, because you now have the ability of these cells to undergo high proliferation. The eutopic endometrium is an estrogenic state, which also has aberrant proliferation. And because progesterone is not able to signal appropriately, you potentially have the situation of infertility. Now, is this relevant to the human situation? So in collaboration with our colleagues in Brazil, we've been able to get some samples from women that have deep infiltrating disease, and essentially looked at the control utopic endometrium, and in the same group of animals, same group of women, before and after surgery for deep infiltrating disease. And you can see here, and these are taken in the mid-secretory phase, and you can see in the women preoperatively that you see an increase in 29C and a decrease in FKBP4. And 5 of these women got pregnant with assisted reproductive therapies after surgery, and 4 of them that did not in the 6 months after the surgery that we were able to then get endometrial tissue. You can see that post-surgery in these women that now 29C has gone down and FKBP4 has gone up. We followed these women, these 4 women that didn't get pregnant in the 6 months, and now 3 of them have also got pregnant. Only 1 of these 9 women have not got pregnant after the surgical responsiveness. So suggesting again that perhaps surgical intervention is responsible for removing the inside of the lesions, and there's a short period of window of time that these women can get pregnant. So now I'm just going to switch to 2021. I know I'm going through this pretty quickly. There's a lot of data here that I want to get through, but I just want to give you an idea of how we approach this process. So microRNA 21, again, is highly upregulated in the eutopic and ectopic tissue of baboons with this disease. And you can, again, see by in situ hybridization, here are lesions taken from baboons at 15 months. And you can see the beautiful in situ hybridization of microRNA 21, both in the glands and also in a subset of immune cells that are found associated with these lesions. And microRNA 21 is really a double-edged sword. It's been extensively studied, like I said, in the breast cancer literature, and essentially it's regulated by inflammation, a response that is critical for endometriosis, and essentially it contributes to fibrosis, it contributes to angiogenesis, and it contributes to reduced apoptosis, all which are critical for maintaining the lesions and things that we need to target if you're going to be able to suppress lesion development. And if you then look to see whether there's a correlation, because IL-6 through STAT3 regulates microRNA 21, you can see that if you look in lesions with for IL-6, you see a significant increase in the messenger RNA for IL-6. And if you take cell lines, whether they are stromal cell lines or epithelial cell lines, and you now treat them with IL-6, you see also a significant increase in the expression of microRNA 21, suggesting perhaps that the inflammatory response that you see in the peritoneal cavity, in the lesions, are important for driving the upregulation of microRNA 21. So to ask whether IL-6 has a direct regulatory mechanism in terms of regulating lesion development, we've developed a mouse model, and in this case what we use is that this mouse model has a progesterone receptor Cree with a ROSA MTMG locus. So we have both coloring, we can red and green fluorescence that we can then monitor using in vivo imaging to see what happens with lesion development. And in this case, all we've simply done is created lesions in these animals using their own endometrium. And as you can see, when we then treat them with IL-6, there's a significant increase in response to IL-6 treatment in lesion development. So this is a very, very nice model system. We have to use model systems. The mouse doesn't get spontaneous disease. but being able to engineer these animals with transgenic technologies, we can then utilize various color mechanisms to try to see whether we can do in vivo imaging. So this really helps us in terms of trying to look at lesion development, trying to understand. And one of the nice things about this animal model is when we looked at them 3 months after we've created the lesions, independent of treating them with IL-6, they become infertile. So basically what you're seeing is an effect that's associated with this. So very quickly then, going back to then ask the question, since 21C is also involved in both EMT and fibrosis, if you now look at EMT in our baboon model at 6 and 15 months, you can see that there is a beautiful upregulation of EMT going on, and these are vimentin positive cells within the epithelial cells of the lesion, and you can see that these cells are trying to transform into a stromal fibroblast, and that's associated with an increase in TGF-beta and phosphosphat, which is involved with the process of EMT. In addition, then if you look again through the developmental processes of lesion development in our baboon model, you can see that if you look at the extent of fibrosis, there is a significant increase in alpha SMA, which is the myofibroblast differentiation associated fibrosis. And then if you stain with Mason trichrome, you can also see the significant increase in the amount of collagen deposition associated with that, which is also associated with the increase in microRNA 21. So quickly, since I need to finish, basically what we see is that the increased expression of 29C and 21 through progesterone resistance and inflammation plays a significant role in the development of fibrosis that's associated with lesion development, and also downstream with the regulation of phosphosphat 3 enhances both growth and angiogenesis associated with the development of this disease. So finally, just to give you a snapshot of what we think might be happening, the very, very early insult of the presence of lesions that we see in our baboon model alters the steroid hormone receptor profile. And again, I haven't gotten into the epigenetic changes that are involved in response to this, which also epigenetically modifies the genome and results in altered expression of microRNAs. So this then results in aberrant gene expression, which can feed back to bring about aberrant proliferation, EMT, and fibrosis to sustain lesion development in these animals. And downstream, the progesterone resistance inhibits the initiation of destrualization, impairs the ability of these cells to destrualize, and as a result of the destrualization defects, this endometrium and the progesterone resistance, this endometrium is no longer responsive to the embryonic signals that are so critical for the establishment of pregnancy. So I've just sort of given you a snapshot, and a lot of our work now is focused on trying to look at the mechanisms associated with this and whether we can target these to try to reverse some of these processes using all of our animal models. So with that, first and foremost, I really need to thank the people that did the work. Oops, go back here, sorry. So all of the microRNA work is being done by Niraj Joshi, who's standing somewhere right there in the back. I haven't talked about any of the work that Mike Strug and Renvi Su have done with regards to any of the notch regulation. Marco Woodson is really responsible for all the animal work we do, and Shara and Samantha are involved with all of the histology and all of the immunohistochemistry that we do. And then I have a wonderful group of collaborators all over the world that have really helped us, and this is not a one-person job. It really needs, it really takes a global village to try to do this. So thank you very much.